Hello and welcome to our presentation. My name is Renee Pinto and my colleagues are Lexan Ambrose and Durrell Nangle. We have selected a paper entitled Social Regulation of Gene Expression in Human Leukocytes for our presentation. The research paper was published on the National Institute of Health website by UCLA, University of California in Los Angeles School of Medicine. We chose the said topic because it was a very interesting topic and appeared to be a popular subject among scientists. It explained a surprising phenomenon of how subjective social isolation is associated with increased inflammatory diseases.
Experiences in daily life can come in many different forms, from feeling socially connected and physically safe to feeling socially isolated and rejected. People who are socially isolated have increased risks of all-cause mortality and several specific infectious and cardiovascular diseases. The biological basis for these epidemiological findings is poorly understood, in part because it is unclear whether the effects of loneliness stem predominantly from the lack of instrumental social support such as physical or cognitive assistance or from the biological consequences of loneliness. The paper sets out to thoroughly explore the adrenal glucocorticoid, which regulates a wide variety of physiological processes via nuclear hormone receptor mediated control of gene transcription.
Cortisol is the most potent glucocorticoid produced by the human adrenal glands. Derived from cholesterol, its production is stimulated by the pituitary adrenocorticotropic hormone, or ACTH for short, which is regulated by corticotropin releasing factor, CRF. Cortisol affects numerous physiological systems, including immune function, glucose counterregulation, vascular tone, and bone metabolism. Cortisol has multiple actions, most notably enhancing the physiological response to stress. Under very stressful conditions, including exercise, trauma, anxiety, isolation, and depression, cortisol levels rise, leading to a chain of events that ultimately provides immediate energy to the body and keeps the individual alert via stimulation of the adrenergic system, the typical fight or flight response. Cortisol activation of the glucocorticoid receptor, GR, exerts broad anti-inflammatory effects by inhibiting nuclear factor, transcription factors, and other pro-inflammatory signaling pathways. However, increased cortisol levels in chronically lonely individuals is paradoxical in light of the fact that most isolation-linked diseases are driven by increased inflammation, for example, atherosclerosis and solid tissue malignancies.
Socio-environmental processes regulate human gene expression by activating central nervous system processes that subsequently influence hormone and neurotransmitter activity in the periphery of the body. Peripheral signaling molecules interact with cellular receptors to activate transcription factors, which bind to characteristic DNA motifs in gene promoters to initiate or repress gene expression. Only genes that are transcribed into RNA actually influence health and behavioral phenotypes. Individual difference in promoter DNA sequence can affect the binding of transcription factors and thereby influence the sensitivity of genomic response to socio-environmental conditions.
Over a period of 5 years, a cohort study took place to assess the psychological, social, economic, and biomedical outcomes of 230 participants. Full medical histories were collected with additional questions on lifestyle choices such as sleep quality and alcohol consumption. By the end of the 5-year period, 166 participants remained out of the 230. This is roughly 72% of the sample size. 11 of these 166 participants weren't willing to have a 10 milliliter sample of blood taken for gene expression analysis, which only equates to 8%. Social isolation was assessed using the UCLAR Loneliness Scale, a yearly questionnaire of answers on a scale from O, I often feel this way, to N, I never feel this way. After the years 1, 2, and 3, 6 samples yielded insufficient RNA for their assays and so were removed, with the 10 top and bottom most consistent results. Background characteristics were assessed using further questionnaires to provide a scale of stress, depression, and hostility furthermore. Blood was collected through venipuncture and 10 million leukocytes were extracted from a 5-fold density gradient centrifugation. 5 micrograms was used in a DNA microarray. Multi-array averaging was used as oligonucleotides were synthesized to detect RNA at a frequency from 1 in 300,000. 22,283 transcripts were assayed and cross-referenced with various databases. Cortisol samples were collected 3 times a day, upon waking, half an hour later, and then 15.5 hours later before sleep, for 3 consecutive days each year. These samples were analyzed using a radioimmunoassay.
Social isolation in human leukocyte is being shown by using transcriptional fingerprint. This is done showing the individual who has high level of loneliness by the intensity of green, which indicates the magnitude of genes relative. And relative under pressure is being shown by using the intensity of red, which is denoted the magnitude. Figure 2, transcriptional control pathway, corrosive level. It was found that there is no significant reduction in average daily saliva cortisol level in high lonely individuals. On the other hand, they did show that they have less diurnal decline in circulating cortisol level than did lonely individuals. The reason for the difference is because even the level of cortisol is high and the change in saliva cortisol concentration is from 0 to 0.5 hours. Post-awakening did not change significantly across groups, but 0 to 16 hours and 0.5 to 16 hours declines were both less pronounced in high lonely individuals.
The paper addresses how a interpersonal world exerts biologically significant effects on the molecular composition of the human body. The paper goes on to show how the genome-wide transcriptional activity in people who are chronically social isolates has elevated cortisol level and in turn has inflammatory diseases. Chronically lonely people with elevated cortisol level should be relatively protected from inflammatory mediated diseases rather than having the increased risk empirically observed. As a result of this paradox, a number of hypotheses were tested to determine what the increased transcription of immune activation genes in lonely individuals might stem from. This study provided the first systematic analysis of genome-wide transcriptional alterations as a potential mechanism of social epidemiological influences on human health. Further contributions of the paper include the use of the structure-function bioinformatics to identify candidate upstream transcription control pathways that drive in vivo functional genomic alterations that are associated with social risk factors. The study also identified a plausible neuroendocrine mediator of transcriptional alteration, which is known to relate to social phenotypes in humans and is causally impacted by experimental social isolation in animal models, is consistent with social influences on gene expression. This study has identified a clear genomic fingerprint of social isolation and defined candidate transcriptional control pathways that may shape its expression, but limitations such as a small sample size must be considered when interpreting these results and thus require replication in larger samples that are more broadly representative of the total variation in human social phenotypes. These data provide the first fact that social environmental risk factors are linked to global modification in human gene transcription, and they ascertain a molecular context for understanding the increased risk of inflammatory disease observed in chronically lonely people. These are references. Thank you for listening.